This week we are starting on chapter 17, the processes of evolution. In our last chapter, chapter 16, we discussed a lot about the history of evolution and how Darwin's theory of natural selection came to be, as well as the evidence that stands for evolution. In this chapter, we'll look more at the different processes that are going on and how that impacts us today. The beginning of our book discusses quite an interesting story, the rise of the super rats. In many places around the world, rats are a big problem. I was amazed to see, and again this is on page 257, I was amazed to see that rats cost us about $19 billion per year. That's something that I actually didn't know. But they ruin crops and they ruin many other things. They bring disease. So previously, back in the 50s, we started using warfarin, which interferes with blood clotting. And it's a chemical. And at the beginning, yes, it was very effective. Rats would die within days of internal bleeding. But as evolution goes, it kind of starts with variation. There's variation in a population, and it turns out that there's some rats carrying alleles that make them resistant to the chemicals in warfarin. And if the other rats without those alleles or genes die, that leaves more resources available for the ones who are carrying the resistant gene. And so they flourish, that gene becomes adaptive, allowing them to survive and reproduce better than the others. And you see that gene, or that allele, which is a version of a gene, increase within the population, and then warfarin doesn't work as well as it once did. That's evolution by natural selection, a classic example and one that affects us today, which is pretty interesting. Evolution starts with mutations in individuals. A mutation is an area of a gene that is different or changed from what might be considered the normal gene, and that introduces new alleles or new genes into the population. Sexual reproduction can quickly spread that mutation through a population. And I'll remind you that a population is a group of organisms of the same species in a specific location who are able to interbreed with each other and create viable fertile offspring. The mutations then create variations in the population. A trait with only two forms is called dimorphic, and traits with more than two distinct forms are polymorphic. Traits that vary continuously often arise by interactions among alleles of several genes and may be influenced by environmental factors as well. Since we have learned that evolution starts with natural selection acting on the variation in a population, here's a list of some things that actually cause variation, starting with, again, the mutations, which is a change in a gene, and that's going to introduce new genes or new alleles into the population. A few other ones that we'll talk about here. Crossing over, when gametes or sperm and egg are being created, chromosomes line up in the center, and then they actually swap big pieces of genes. Independent assortment has to do with how, when sperm and egg are being made, how the chromosomes line up on either side of the cell to be prepared to be split apart into separate cells. And remembering that for every chromosome you have, you get one from your mom and one from your dad, with a total of 23 pairs of them, and what side they line up on, it helps to mix up the genes that you get from your mom and dad into separate sperm or separate egg, depending on if you're male or female. Fertilization then with a new mate restores the chromosome number from 23 to 46, creates new alleles, combines alleles from the two parents. That helps bring about more variation and is the definition, the wonder of sexual reproduction. Also changes in chromosomes, which is a little bit different than a mutation in that pieces of a chromosome might be lost or they might be duplicated and transposed onto another chromosome. All of those are sources of variation. Mutations are the original source of new alleles. Some can be lethal or deadly, some can be neutral. If they're lethal, they could cause death right away or soon thereafter. Phenotype here is a physical characteristic, so a mutation that drastically alters a phenotype, which can cause death. Neutral is one you don't even notice, so there's no effect on survival or reproduction. Occasionally, the environment will favor a mutation. It will help that individual survive better and therefore reproduce and pass on that mutation to its offspring. Mutations are the source of the Earth's staggering biodiversity. All alleles in a population, or all genes in a population, form a gene pool. If you imagine a population with all the individuals and all the genes that each of them have, kind of like a little pool of genes that are available within that population. Microevolution is when there's changes in the allele or the gene frequencies of a population, and occur constantly by processes of mutation, natural selection, genetic drift, and gene flow. We'll talk about each of those separately. Again, the gene pool is all of the genes or alleles in a population, like a pool of genetic resources. Microevolution, which is evolution on a small scale, is a change in allele frequencies in a population over time. It may not bring about new species right away, but over time, when added up, can result in new species. The allele frequency is the abundance of a particular allele among members of a population. A theoretical reference point, also called a genetic equilibrium, occurs when the allele frequencies of a population do not change. It would require 5 conditions in order for a population not to change. In nature, it seems that those conditions are never quite met, all 5 at a time, so natural populations are never in genetic equilibrium. Here's a list of the 5 theoretical conditions of genetic equilibrium. That mutations never occur, population is infinitely large. That means you have a large gene pool to pick from, plenty of genes available. The population is isolated from all other populations of the species, so no gene flow like migrations of other individuals from other populations infiltrating your population. Mating is random, meaning there's no special sexual selection going on for certain traits, that it's just all random, and that all individuals survive and reproduce the same number of offspring, so no natural selection. Seeing this list really does show that nature cannot be in genetic equilibrium, because genes are indeed continually mutating, and it's very often that sexual selection occurs as well as natural selection. Microevolution: individuals of a population inherit different alleles, and so they differ in phenotype. Again, a phenotype is a physical characteristic, and an allele is a version of a gene. For instance, blood type comes in 3 alleles: A, B, and O. That's a good example of alleles. Over generations, any allele may increase or decrease in frequency in a population, and such a change is called microevolution, evolution on a small scale. Researchers know whether a population is evolving by tracking the deviations from a baseline of genetic equilibrium. So using that previous list of when a population's in genetic equilibrium as their baseline, to know when a population wouldn't be evolving, they can track the deviations from that to know how a population is indeed evolving. The whole phenomena of doing that was first thought up by a scientist team, Hardy and Weinberg. Now it's called the Hardy-Weinberg formula. Gene pools can remain stable only when those 5 theoretical conditions of genetic equilibrium are being met. So Hardy and Weinberg developed a simple formula that can be used to track whether a population of any sexually reproducing species is in a state of genetic equilibrium. And the following illustrates how the Hardy-Weinberg formula is used. So consider a hypothetical gene that encodes a blue pigment in butterflies. 2 alleles, or 2 genes of this allele, big B and little b, are codominant. So remember, for sexually reproducing organisms, you get, for each trait, 1 gene from mom and 1 gene from dad. So this butterfly here would get a big B from mom and a big B from dad, and would be dark blue. A butterfly homozygous for the little b allele would get a little b from mom and a little b from dad, and would have white wings. When they're homozygous, it means they get the same allele from each parent. But when you're heterozygous, you get one of each of the alleles. From one of the parents, they gave you the big B, and the other parent gave you the little b, which creates kind of an in-between, what's called codominant situation, where you would have medium blue wings. At genetic equilibrium, the proportions of the wing color genotypes are as follows in this equation right here. And they should add up to the frequency of 1. P is just used to symbolize the frequency of the alleles. So P squared is when your genetic combination is big B, big B. Q squared over here is when your genetic combination is little b, little b. So we saw this one over here would be blue, and this one here would be white. And the in-between alleles, for medium blue color, the heterozygotes, they're 2PQ. They arrive at that because there's 2 different ways to get the heterozygote condition. Dad could give you the big B, mom could give you little b, or vice versa. Mom gives you the big B, dad gives you the little b. So 2 ways to arrive at that, so 2PQ. So adding up the frequency of the big B, big B's, plus the heterozygotes, plus the little b should give you a frequency of 1. So we can use a little algebra here. If we know one of these, we can figure out what the other is. And scientists can count alleles in a population, and that's good if you're in needing of knowing something like how many disease alleles are in a population. The frequencies of just the big B and the little b should add up to 1 as well. So P plus Q is 1. So back here, these were the genotypes, those adding up to 1. And just the alleles that are in the population should add up to 1. So the amount of big B's added to the amount of little b's, P plus Q, should equal 1. Example, if big B occupies 90% of the loci, that's the location of that gene on a chromosome, then little b should occupy the remaining 10%. We can do a little math, turn those percents into a decimal, and they should add to 1. And here's an example of a problem. At fertilization, gametes combine at random and start a new generation. If the population size stays constant at 1,000, there will be 490 big B's, big B combination genotypes. That's called the homozygous dominant genotype. 420 heterozygotes, big B, little b, and 90 homozygous recessive individuals, little b, little b. And all those add up to 1,000. If we were to plug this into the Hardy-Weinberg equation, such as back here, remember those frequencies add up to 1. All allele frequencies for dark blue, medium blue, and white wings are the same as they were in the original gametes, then the population is not evolving. In the real world, researchers can use the Hardy-Weinberg formula to estimate the frequency of carrier of alleles that cause genetic traits and disorders, such as your book's example of the hereditary hematochromatosis, which is in Ireland. It's kind of nice for doctors to be able to calculate how many people they can expect to have these conditions. Patterns of natural selection. Natural selection occurs in 3 different patterns, depending on the organisms involved and, of course, their environment. Remember, natural selection is the process in which environmental pressures result in differential survival and reproduction of individuals of a population who will vary in details of shared heritable characteristics. So, in other words, organisms that are better adapted to their environment will survive and reproduce. And here's our 3 examples of natural selection. They are directional selection, stabilizing selection, and disruptive selection. And I'm going to flip to another screen here to show you pictures of those to visualize them a little bit better. Alright, here is a slide showing our 3 types of natural selection. We look up here in the graph, and let's say this is our original population of mice, and we're looking at fur color here. Variations include the white down here to the very dark up on this end, with your average being the brown in the middle. Now, if this was directional selection, our graph would shift one way or the other. In this case, it's shifting more towards the darker end of the variation, so that again, the dark red line is our original population, and the dotted line is the new population. We've had a directional shift in this phenotype or this physical characteristic. That could be because natural selection is selecting for a darker mouse. Maybe the background that they live in is darker and they can hide better from predators if they have a darker coat. And the white ones get picked off easier because they show up to predators better. If it's disruptive selection, if we see this pattern happening again, the dark red line is our original population and the dotted line is the new. Our two ends are selected for. That the white do well and the very dark do well, but the ones in the middle, the brown, do not. And in the end here in graph C, we see that our intermediate area, our middle area, is selected for the brown, and it looks like our population of white and really dark ones decrease so much that our graph kind of skinnies out, so that most of our mice are indeed brown. Natural selection has selected for just the brown without too much variation on the ends. Again, these are the three types of natural selection: directional, disruptive, because it disrupts our mean here, and then stabilizing, because it really stabilizes the middle. So you can page through, and your notes will talk about each of those as I just did, while looking at that other slide with the pictures on it that your book did not have. And they give their own examples of each in the book, from directional selection and the peppered moth, the rock pocket mice, and antibiotic resistance. Stabilizing and disruptive selection. Definitions once again of each of those. And the example of the sociable weavers for those. Disruptive, and the African seed cracker examples. And you can check your book for those examples there. Fostering diversity. Selection pressures that operate on natural populations are complex. An allele may be adaptive in one circumstance but harmful in another. Any mode of natural selection may maintain two or more alleles in a population. With sexual selection, also called non-random mating, the most adaptive forms of a trait are those that help individuals defeat same-sex rivals for mates, or are the ones most attractive to the opposite sex. Perhaps what the females might be choosing when they choose their mates. Sexual selection is the mode of natural selection in which some individuals of a population out-reproduce others because they're better at securing or getting mates. There's some trait they have that allows them to mate more often and more successfully, and therefore have more offspring. For example, male elephant seals fight for sexual access to a cluster of females. A male bird of paradise engages in flashy courtship display to catch the sexual interest of a female. Females are choosy. A male mates with any female that accepts him, but it seems that females are the ones that are actually doing the careful selection. They only have so many offspring at a time because of limited amounts of eggs, but with males there's a lot of sperm, and so they're not too choosy. So it seems sexual selection is most likely driven more by the female, by her choice. Stalk-eyed flies cluster on aerial roots to mate. Females prefer males with longer eye stalks. So a male with very long eye stalks has captured the interest of 3 females, and those pictures are in the book. So you can see over time, with females preferring males with longer eye stalks, how those alleles would become more prominent in the population. An example of microevolution. Balanced polymorphism in an environment that favors heterozygotes, or individuals with non-identical alleles. Any mode of natural selection may result in balanced polymorphism. Balanced polymorphism is the maintenance of 2 or more alleles for a trait at high frequency in a population as a result of natural selection against homozygotes, or those having the same allele, like big B or little b. So heterozygotes would be like the organisms who have big B and little b. For instance, great example is the malaria and the sickle cell anemia. There's a mutation in hemoglobin, and if you carry this mutation, you have what's called sickle cell anemia. Individuals homozygous for the mutated allele often die young because this mutated gene causes a malformed protein that doesn't work as well as hemoglobin to carry oxygen on red blood cells. But this allele persists at high frequencies in tropical regions of Africa, because if you're heterozygous and you only have 1 copy of the bad allele, you're actually more likely to survive than those that are homozygous dominant, who have both normal alleles for hemoglobin. The reason being is that having 1 of each copy seems to help flush the malaria protist out of your system faster than if you were the homozygous dominant combination. But then again, if you have an area that has both the heterozygous and the homozygous dominant, when they get together every now and then, they will have an individual or an offspring that has both the bad alleles and then actually has sickle cell anemia, and then they would die young. Genetic drift is something that also causes variation in a population. A random change in allele frequencies or genetic drift can lead to loss of genetic diversity by causing alleles to become fixed or lost in small populations especially. Genetic drift is a change in allele frequencies in a population due to chance alone. They're random. Fixed refers to an allele for which all members of a population are homozygous, so no heterozygotes for that particular allele combination. So either all big B's or all little b's. The larger the population, the smaller the impact of random changes in allele frequencies. For instance, if you have a population of 10 and only 1 person has that allele, that 1 person dies, the allele is lost forever. But in a population of 100, all 10 people who carry the allele would have to die before the allele would be lost. An example of genetic drift is bottleneck. This is when it can be very dramatic when a few individuals rebuild a population or start a new one, such as hunting reduced an elephant seal population down to 20. The population is now homozygous at every gene. Genetic drift after the bottleneck fixed all alleles in the population. Cheetahs are a good example of organisms that went through a bottleneck in the last ice age 10,000 years ago, and then again with hunting and poaching. So much so that they've lost a lot of their variation in their population, and each cheetah is very extremely, almost genetically identical. Variation in genes is good because if organisms have varied genes, if bacteria or viruses come along, they're much more likely to have a survivor to help repopulate the species. Founder effect is another example of genetic drift. For instance, if, let's say, imagine up in the corner here we've got these pictures of these birds for founder effect, that in the original population you have both red and yellow alleles, but should a piece of that island theoretically break off, sounds silly, but it actually does happen, maybe only the red ones were on that slice of land and therefore ended up on that floating island, and so now the yellow allele is lost. So these new individuals inhabiting the new area are the founders, which you're going to only have just their gene pool, not the original gene pool. So again, that's called founder effect, and that is a type of genetic drift. It's a chance event that causes variation and then evolution. Inbreeding. Genetic drift is pronounced in inbreeding populations. Inbreeding lowers a population's genetic diversity, so more individuals in the population are homozygous for recessive alleles with harmful effects. For instance, the disease Ellis-Van Creveld syndrome, which the symptoms are dwarfism, polydactyly, that's extra digits or fingers and toes, and then also, unfortunately, heart defects, which was prominently found in the old order Amish of Lancaster County, Pennsylvania. And that was a result of inbreeding. Gene flow. Individuals with their genes, with their alleles, move into and out of populations. Like, for instance, migrating, emigrating, immigrating to areas. Think of yourself like a little package of genes, and when you go somewhere, you carry your genes with you. And perhaps if you go to a new area, to a new population, perhaps the genes you're carrying could be a little different than the ones that were in the area that you arrive at. That's gene flow. Here's an interesting slide I have that's extra on gene flow. And we can kind of see the results of it with humans, with our ability to travel anywhere and everywhere. That with our transferring of alleles between populations of humans, we ask, are we moving towards a blended world? This was an article in Time magazine at one time with a computer-generated model of what could be the new face of America when we mix all of our genes because of gene flow. I thought that was kind of interesting. Here's another example of gene flow, how blue jays move acorns and their alleles among populations of oak trees that would otherwise be genetically isolated. Key concepts: the processes of microevolution. Natural selection may maintain or shift the range of variation of a shared heritable trait in a population. Gene flow counters the evolutionary effects of mutation, natural selection, and genetic drift. Reproductive isolation. Speciation, which is the development of new species, differs in its details, but reproductive isolating mechanisms are always part of the process. So reproductive isolation is part of the creation of new species. when gene flow doesn't occur between populations, different genetic changes accumulate in each population. Reproductive isolation reinforces differences between diverging populations. For instance, if pollination or mating cannot occur, or if zygotes cannot form, the isolation is prezygotic. Zygote refers to after fertilization by sperm and egg, the ball of developing cells. So if you can't get fertilization to happen, such as in pollination, or actual fertilization between sexually reproducing organisms, it's called prezygotic. If hybrids form, so you actually do get two species to reproduce with each other, but those hybrids, that's what they're called, are unfit or infertile, the isolation is post-zygotic, meaning you were able to have fertilization happen and you've created a zygote. Perhaps even the zygote was born, but it's infertile, like in the case of a mule. Or unfit, meaning it can't even survive, it's post-zygotic isolation. Alright, we're going to go through some types of isolation here. Some of these will be prezygotic and some will be post-zygotic. And I'm going to flip to some different slides here. I'm going to diverge off of our book's PowerPoint. This picture is courtesy of Pearson Education textbooks, and I like it. It shows us a range of types of barriers that keep individuals from reproducing with each other, and therefore when enough differences build up, new species are created. So we're going to start down here with habitat isolation. Habitat isolation is when two species encounter each other rarely because they're in different areas, such as these garter snakes. One lives in dry land and one lives in the water. And again, these pictures are courtesy of Pearson Education. Thank you to Pearson. They will never meet up with each other, and therefore they won't have gene flow. Their gene pools will become isolated, and new species will arise. When they finally do meet up again, they'll be too different in order to reproduce with each other, so new species will occur. Temporal isolation, those are species that breed at different times of the day, different seasons, different times of the year, and therefore if they're not feeling in a mating type of way at a certain time as someone else, they won't get together to mate, such as these types of spotted skunks that one mates in the fall, one in the spring. So as time goes on, they will become separate species because they won't mix their gene pools anymore. Behavioral isolation, such as courtship rituals and other behaviors that are unique to the species, provide effective barriers, such as these blue-footed boobies recognize each other by a courtship dance. If the dance isn't done just right, or if behaviors aren't just right, they won't recognize each other as part of their species, and they'll stop mating and therefore stop mixing their gene pools. Gene flow is reduced and new species arise. Mechanical isolation is when there's changes in structures that are too great that prevent successful mating, such as these snails. Their reproductive parts don't line up quite right. One's down here, one's over there, and they're not going to be able to get the job done to transfer sperm. Therefore, fertilization won't happen. Gametic isolation, when the sperm of one species is not able to fertilize the eggs of another, probably because the proteins on the cell surfaces don't match up. like the sea urchins are all sending their sperm and eggs into the same pool here, but if the proteins don't match up, they won't fertilize. So luckily in this case, the species, the ones that are alike, their sperm will find their correct eggs. Again, now some post-zygotic barriers, such as reduced hybrid viability, where you actually do get together, everything's okay, you form a zygote, but you make a baby, but the baby, called a hybrid because it's between species, is so weak that it barely survives and then dies an early death before it's able to reproduce, such as the salamander. Reduced hybrid fertility, so you make a baby, which is again called the hybrid, and they're actually quite vigorous and healthy, but the baby is sterile, such as the example of the mule. Hybrid breakdown, so that takes the mule a step further. This is common in plants, where you actually make a very viable offspring that's able to survive, maybe even into the next generation, but they break down after that, become feeble or sterile later on. This is all part of what's called the biological species concept, in how we define a species as being able to get together and mate and create viable, fertile offspring. Otherwise, you would be considered a different species. Again, thanks to Pearson Education for those pictures. Some other types of speciation: allopatric speciation, when a geographic barrier arises and separates two populations. Over time, mutations can build up, and when there's no gene flow between the two populations, we can get new species arising from that. Again, allopatric, you have a geographic barrier, such as the example of the Isthmus of Panama, cut off gene flow among populations of aquatic organisms in the Pacific and Atlantic Oceans. Such as also the speciation in archipelagos, such as the Hawaiian and Galapagos Islands. They get populated by just a few individuals from the mainland, which then diverge into new species over time, filling new niches and habitats, having adaptive evolution occur. The Hawaiian honey creepers, that example is from your book. Sympatric speciation, so your population is still near each other and even in physical contact with one another, but there's some sort of physical barrier, that was allopatric, but something that bars them from mating with one another. Such as a chromosome number change, something usually with sympatric, it's something genetic or chromosomal. And this does actually often occur with plants such as wheat. So again, sympatric speciation is a pattern in which populations inhabiting the same geographic region speciate or change into new species in the absence of a physical barrier between them, because it's more of a genetically isolating thing, such as having too many chromosomes. Organisms need to have, if they're the same species and able to mate, need to have the same amount of chromosomes. Like the Lake Victoria cichlids and the warblers on the Tibetan Plateau. Check out your book for those examples. Parapatric speciation can occur when one population extends across a broad region with diverse habitats, like the two species of velvet walking worms with overlapping habitats in Tasmania, where they interbreed and their hybrids are sterile. Parapatric speciation, this model in which different selection pressures lead to divergences in a single population. New species again arise. It always involves the end of gene flow between the populations. So it isolates the two populations, and mutations and such build up, so that when you finally do reintroduce the two populations, they're too different to be able to reproduce anymore. Hence, new species have arisen. This is microevolution. Macroevolution, this is evolution on a larger scale, and this is the buildup of many microevolution events over time. A central theme of macroevolution is that major evolutionary novelties often stem from the adaptation of an existing structure for a completely different purpose, or exaptation. This includes, macroevolution includes patterns among or above the species level, giving rise to multiple species, origin of major groups, and even in major extinction events. 4 patterns of macroevolution emerge: stasis, adaptive radiation, coevolution, and extinction. With the simplest macroevolutionary pattern, stasis, a lineage persists for millions of years with little or no change, like the coelacanths. Stasis means staying the same. Mass extinctions, more than 99% of all species that ever lived are now extinct. It seems that there have been more than 20 mass extinctions, which are simultaneous losses of many lineages, including 5 catastrophic events in which the majority of species on Earth disappeared. Extinct refers to a species that's been permanently lost. In adaptive radiation, a lineage rapidly diversifies into several new species. It can occur when individuals colonize a new environment that's got a variety of different habitats with no competitors, like, for example, the Hawaiian honeycreepers. It's like a burst of genetic divergences from a lineage, giving rise to many new species. It can also occur after a geologic or climatic event, which would eliminate competition from a habitat, allowing others to move in, such as when the dinosaurs disappeared, allowing mammals to undergo an adaptive radiation event. A key innovation can result in adaptive radiation or rapid diversification into new species. For example, the evolution of lungs opened the way for an adaptive radiation of vertebrates onto land. A key innovation is an evolutionary adaptation that gives its bearer the opportunity to exploit a particular environment more efficiently or in a new way. In coevolution, close ecological interactions between two species cause them to evolve jointly. Over time, they can become so interdependent that they can no longer survive without one another, such as the large blue butterfly and the red ant. I'm going to pull up another picture. Here's a nice picture of coevolution, for example, cheetahs and gazelles. How nature has created such fast and alert animals. With the hunting continually of gazelles, they've become faster, as cheetahs have to become faster to be able to catch their prime food source. Great example of coevolution. How hummingbirds' beaks grow longer to fit their flower to get nectar, or why leaves on a tree are so high with the giraffes' necks. Although there's more to the giraffes' necks than just that. There's also their mating fights, I guess you could say, where they knock necks with each other. So, coevolution is a joint evolution of two closely Interacting species. Each species is a selective agent for traits of the other, and each adapts to changes in the other. In your book, they give you example of the ants and the butterfly. Take a look at that. Evolutionary theory. Many biologists disagree about how macroevolution occurs. Dramatic jumps in morphology may be the result of mutations in homeotic or other regulatory genes. Macroevolution may be an accumulation of many microevolutionary events, or it may be an entirely different process. Instead of trying to divide the diversity of living organisms into a series of taxonomic ranks, many biologists are now focusing on evolutionary connections. Cladistics allows us to reconstruct evolutionary history, which is called phylogeny, by grouping species on the basis of their shared characters. The result of a cladistic analysis is a cladogram, which is a type of evolutionary tree where you can group individuals according to their common ancestor and their shared traits. Every branch ends with a clade, or a species or group based on a set of shared characters. Ideally, each clade is a mono, that means one monophyletic group that comprises an ancestor and all of its descendants. Sister groups, the two lineages that emerged from a node on a cladogram, and a monophyletic group is an ancestor and all of its descendants, such as the Hawaiian honey creepers, which are given example in your book. Isolation that spurred honey creepers' adaptive radiations also ensured they had no built-in defenses against predators or diseases from the mainland. Specializations became hindrances when habitat suddenly changed or disappeared, and at least 43 species of the honey creeper that thrived on the islands before humans arrived were extinct by 1778. Today, 32 of the remaining 71 species are endangered and 26 are extinct. Destruction of food sources and avian malaria decimated the Palea and Akiki, I'm not sure how that's pronounced, and the Puli are probably now extinct. There's a picture of those birds in your book. Cladistics uses evolutionary tree diagrams. They're based on the premise that all species interconnect through shared ancestors. Grouping species by shared ancestry better reflects evolutionary history than do traditional ranking systems. And then in the end, we revisit the rise of the super rats. It's interesting that the allele that makes rats resistant to warfarin is adaptive when the warfarin is present, but when it's not present, when you've got the others who don't have the allele competing with you, it's actually maladaptive. The periodic exposure to warfarin maintains a balanced polymorphism of the resistant gene in rat populations. Courtesy of a picture of Pearson, I wanted to show you an example of a phylogenetic tree, also called an evolutionary tree. This area here would be the common ancestor to all of these organisms here. All of these organisms are still alive today because the present line is right here. Let's say the lancelet line only came to here. That would mean that it would be extinct today. But it's not. It actually makes it up to the line that everybody else does. These are homologies here, or shared traits that show up along the way. So these trees are a nice way to organize this and show when certain traits show up. And it shows who can be more closely related. So obviously a turtle is more closely related to a leopard than it is to a salamander, interestingly enough. Here's another one. Showing some mammals here. The wolf and coyote. Much more close related to each other than to any of these guys over here. Our leopard here would be called the outgroup because it's the least related to all these other four. And again, here is our common ancestor. Again, make sure that you take a look at the questions at the end of the chapter, read the chapter summary, answer those questions, do the critical thinking. That'll help you as well.